Good evening, class. Please let's unmute and respond if you can hear me.
Good evening, ma.
Good evening, ma.
Good evening, ma.
Class.
Evening, mom.
Can we hear me? Okay, you
Yes,
cannot
ma'am.
hear me. Good evening, class. Can you hear me now? I've been talking for some time now. Good evening, class. Please unmute and respond if you can hear me.
Ma, we can hear you.
Okay, thank you. So, decided to start my part of BCH 320 so that we will not have to need to rush towards exams. Who would like to give us a word of prayer so that since it's our first interaction, let me be the one to give the word of prayer. My name is Dr. Talabi. I'm a lecturer in biochemistry department. I don't know how many of you have met me before. Has any of you met me in the department, or did I take you any course in 200 level? Have we had any interactions before now? Hello, are we there?
Yes, ma. BCH 322, ma. BCH 222, ma.
Okay, what's that? What's 222?
Introduction to Biochemistry 2. Say that second semester course.
Okay, so we've met before now. Okay, that's great. And I hope nobody had any carryover in that course. Is
No
that-
idea, ma.
Okay, so good to meet you. So we hand this session over to the Holy Spirit and we ask that He gives us understanding in Jesus' name. Amen. Now I'm going to launch the poll. The poll is the attendance, and I'll just launch it for a few minutes. So once you see it, please fill in your details. That will be the first attendance. The second attendance will be answers into the chat box. When I give it, there'll be quizzes on and off in the course of the lecture. So I'm just trying to let us know how it will go. So there'll be quizzes on and off, and you'll respond by putting your answers in the lecture. I'll also be sharing a YouTube link. The YouTube link is is the recording for the lecture so that you could always go back and listen to it at will. So I will lay down one or two rules for the class. Can we see the material I'm sharing? I'm sharing the Can we see it, please? If you can see it, let me know what you are
Yes,
seeing on
ma'am.
your screen.
Yes, ma'am. I can see.
Can you see the lecture note that is being shared?
Ma, we can
Yes,
see.
ma'am.
What does it say? What are you seeing? Tell me what you are seeing.
Ma, we can see signal transduction, adenylate cyclase system, G proteins, and the rest, ma.
Okay, thank you. So the first topic in my part of the course is signal transduction. And what is signal transduction? Signal transduction is all about transferring signals, just like it sounds, using hormones, neurotransmitters, through receptors. So we said, like, if somebody steps on a pin now, what happens that you can quickly remove your leg because you felt pain in the split of seconds? That's because these receptors on the skin surface quickly send message to the brain, and the brain says, remove your leg from that thing before it causes more damage to the causes more harm to this and breaks the skin. So that is in a layman's language. Now, cells often receive extracellular signals. The cells are inside our body. If you remember the organelles, they often receive extracellular signals through hormone neurotransmitters via the receptors. And the key players to these transducers, that is vehicle, the vehicle for transference of these signals, are what we call G proteins and second messengers. We also have protein kinases, and so we're going one after the other. The topic once again says signal transduction, adenylate cyclase system, G proteins, guanylate cyclase, protein kinases, phosphoproteins, phosphodiesterase. Now, just to freshen our memory, when you see ASE at the end of a word, what does that tell you? What does that tell you that is? If you see ASE at the end of any word, it's what that is an enzyme. You understand? So most of these second messengers are enzymes. They act as enzymes. And you know, if you remember the cliche that was often always said, it says all enzymes are protein, but not all proteins are enzymes. So we're saying that the key players in signal transductions are G protein, second messengers, protein kinases, and enzymes. And all enzymes that synthesize or degrade messengers. Those are the key players in signal, in transference or transmitting signals. So we start with G proteins. What is G protein? G protein is a molecular switch. It acts like a molecular switch. What does a switch? Just like our normal switch in our houses, a switch turns on or off any reaction. So the G protein is a molecular switch, and it connects the G protein coupled receptors, that's the GPCR. Some of you in your 400 level, you'll be working on this G protein in your research. So what does it do? The G protein connects the GPCR on the cell surface to the inner part of the cell. And what does this mean? That means it is one that helps to transmit signals from outside the cell into the cell where the hormones and receptors are for cellular response. Now, this G protein is made up of alpha, beta, and gamma subunits. And each of these subunits has its own function. The alpha unit is the most important because it can bind GDP in active state or GTP in the active state. And what is GDP? GDP is what? Guanosine what? What is GDP? Anybody?
Guanosine diphosphate.
Yes, thank you. Now, the beta and gamma complex, they are often together, and they also send signal in some cases. But like we said, the alpha subunit is the most important in the G protein structure. Now, how does it work? In the resting state, we said G alpha is bound to GDP. That means it's off. And the three subunits are together, and they are all together attached in the inactive form on the GPCR receptors. Now, if G protein is to be activated, that means if a signal is supposed to be sent from outside the cell to the inner part of the cells, a ligand, a signal molecule, or a ligand, or epinephrine, that's a hormone, binds to the GPCR receptor and changes its shape. Now, that tells us that that is an allosteric enzyme. An allosteric enzyme is an enzyme that has a conformational change when a ligand binds to the allosteric site. Now, what happens when this does this is that it causes the G protein to release GDP and pick up GTP, thereby becoming active. And then the third step is the signal transmission. Don't forget, the first step is the resting state, the inactive state. G alpha is bound to GDP. I'm trying to paraphrase to recap. The activation is the um, GPCR binding to its receptor, thereby causing a conformational change and then leading to the activation of G alpha by getting it bound to GTP. Now, signal transmission, the complex now splits. G alpha separates from beta gamma, and then once it is active, this subunit can either activate or inhibit enzymes inside the cell depending on the need of the body at that moment. And once it has elucidated its effect, once it has completed its work, it becomes deactivated again so that it doesn't continue working and then cause harm. That's what happens during cancer. In cancer, the cells just keep on proliferating and proliferating non-stop. So it must become inactive if it has finished its work. Please, is the slide moving as I'm moving, or is still at the title page? Is it moving as I'm
It is
moving?
moving, ma'am. Yes, ma'am.
Okay, thank you. So that is how the G proteins work in four major steps. Now, we said there are different types of G proteins. You have GS, GI, and GQ. GS and each of them are specific for their roles. And these types of G protein are categorized based on their alpha subunits. So GS stimulates adenylate cyclase, and an example is epinephrine. And once this happens, it increases levels of cyclic AMP. CAMP means cyclic AMP. Now, GI functions in the inhibition of adenylate cyclase, which reduces the level of cyclic AMP, while GQ activates phospholipase C, and it works with many hormones. So the most important thing to take away from here is G protein helps in transmission of signals. How is this regulated? Um, in the body, it helps in hormone actions, the physiological roles. Hormone actions either by working with glucagon or epinephrine. Also for vision, there is a special G protein in the eye that is responsible for sight. And then for smell, for heart rate, blood pressure, G protein is implicated in many physiological roles of the body. Now, in a nutshell, the major points to note is that these G proteins are like the on and off switch of the cells on the cell surfaces that are controlled by either GTP or GDP. They allow one receptor to affect many downstream molecules, and then the cycle prevents signal from staying on forever, so that it doesn't behave like a situation in cancer where there's no more, the cell has lost control, it just keeps on growing and reproducing itself. Now there are different types of G protein that leads to different second messengers. Now we go to the next second messengers. All of these second messengers are, what are second messengers? Let me even stop feeding you. Oh yeah, who can tell us what are second messengers? I give everybody 2 minutes. This is 7:22. Type your answer, just the answer, to the in the group chat. That's the first quiz. And please note that this meeting is recorded. So it's that's the second way of seeking active. Let's see, what are second messengers? So answer in the chat box. In the chat box for Zoom, or not WhatsApp, please. In the Zoom chat box. Let's see what. Let me see what we our knowledge. Let me tap from the knowledge. So the first part of that question is, what are second messengers and how are they different from first messengers? How are they different from first messengers? So the two in one question. What are second messengers? What differentiates them from first messengers? We have 1 more minute. I'm looking at the times, and this is timed. So let's see. Second messenger, small intracellular molecules released. Ah, you are busy going to just in your own words. This essay is too long. What are second messengers? How are they different from first messengers? Just a like 2-line answer. It just few words. I don't need essays. They relay messages and amplify sounds received from outside. Now, how are they different from first messengers? Nobody is answering the second part. That's why a lot of you don't. There are no A students in this class. Nobody is even giving example. That's what differentiates the A student. The A student goes the extra mile. You know, example, you're just first messengers outside the cell, start signal. Second messengers are inside the cells. Okay, this is 7:25. So only this amount of people submitted at this time. Okay, so now what are examples of second messengers? Now, the one we are doing now exactly are second messengers. Cyclic KMP, GTP are second messengers. They are inside the cell. They transfer signals inside the cell, while the first messengers are the hormones and the neurotransmitters, and those ones are extracellular inside the cell. So thank you. Back to our lecture. So this next second messenger we're talking about is adenylate cyclase system and its pathway. Just like we defined GTP, what is adenylyl cyclase, also known as AC? They are membrane-bound enzyme. I told you once you see ASE at the end of any word is an enzyme. So adenylyl cyclase are membrane-bound enzymes. They act as key effectors in many signaling pathways. Okay, so ATP, it converts ATP into cyclic AMP, and this is a one-line equation. We can see ATP going to cyclic AMP plus pyrophosphate. Okay, there are different types, isoforms of this adenylyl cyclase. And how does it work? One step is by signal arrival. A hormone binds to the GPCR receptor. When this happens, G protein is activated. We saw how G protein is activated by G alpha binding to GTP. So once G protein is activated, adenylyl cyclase adenylyl cyclase is now activated by when GS alpha, the activated G alpha binds and stimulates adenylyl cyclase to become active. Now, this second messenger production happens very rapidly, and it hydrolyzes ATP by removing, ATP is adenosine triphosphate. So hydrolysis is breaking down. So it removes one phosphate group, and so it becomes cyclic AMP, that's adenosine monophosphate plus inorganic phosphate, and this diffuses through the cytosol. Now, just a minute, please. Okay, thank you for the minute. Now let's continue, please. Where did I stop? Okay, we've said how it works, how it is activated. Now, the main targets for cyclic AMP is the protein kinase A, PKA. And PKA is always in the inactive form. Please note that most of these enzymes, they are produced in the inactive form, are known as zymogens. Zymogen is the inactive form of an enzyme, and it's just for a simple reason, so that they don't damage the body. Because an enzyme, if it is activated, it elucidates its effect and becomes inactive back. Because if it's left to be active, it can, after it has finished working and it continues, keeps on doing what it's not supposed to do, it can cause harm or damage a part of the cell, which could be detrimental over time. So the main target for this cyclic AMP is the PKA, which is also produced in the active form, has a tetramer. A tetramer, talking about 4 units. You have 2 regular subunits and then catalytic, 2 regulatory subunits and then 2 catalytic subunits. Now, talking about enzymes, when you hear of regulatory and catalytic, an enzyme having 2 sites, what does that remind you of? What type of enzyme is that? 300 level biochemistry students. When an enzyme has a regulatory site and a catalytic site, what type of enzyme is that? Yeah, send your answer to the group chat so that I'll not wait forever. You have another 2 minutes. Send your answers to the group chat. An enzyme that has a regulatory site and a catalytic site is known as what? Let's see. You just have 2 minutes, so by 3:30 for our complete class. By the way, if you have not filled your poll, I'm going to take it down now. The time for second messenger has gone. Some people are sending second messenger answers. I need the answer for the what that enzyme is called. An enzyme that has two sites. What is it called? Exactly. Thank you. Allosteric enzymes. Ah, and I told you about A students. A students. Exactly. Thank you. And you not Temitope. An allosteric enzyme is a regulatory enzyme. That's what I was going for. So they always are the regulatory point of any chemical reaction. Now, this pKa, like we were discussing, has both catalytic and regulatory. It tells us that it's a regulatory enzyme. Okay, so the main target for the CAMP is the pKa, and it exists as a tetramer. Now, once CAMP is activated, it binds to the regulatory subunits of the pKa, causing them to release the catalytic subunit. And then once the catalytic subunit is now active, it's the it phosphorylates the target protein on the serine residue, serine or threonine residue. You know, proteins are made up of amino acids. So serine or threonine are types of amino acid. If I'm too fast, please let me know. If I, or if you cannot hear me, let me know. Are we good? Should I continue? Any questions so far before I continue?
No, ma.
Okay, thank you. So we go on to the next, what we have next. Now, what are the downstream effects of pKa? pKa helps in the breakdown of glycogen. When there is no food, you know, glycogen is excess carbohydrate. Is when you overeat, the excess glucose that is broken down, the excess carbohydrates that is broken down is stored as glycogen. Now, in the absence of food, like maybe when you're fasting or during starvation, glycogen is broken down through glycogenolysis. And how does this happen? pKa phosphorylates phosphorylase kinase. And when this happens, once once you hear about any phosphorylation reaction is an activation reaction. So what it's trying to do is to activate glycogen phosphates, which will now start trigger the breakdown of glycogen to glucose, because the glucose is what the body needs to produce ATP. Also, for gene expression, pKa phosphorylates cyclic AMP response element binding protein, that's CREB. And once this is activated, I told us phosphorylation, all phosphorylation reaction are activation, are a way of saying activation. So once CREB is activated, it enters the nucleus and turns on gene transcription. So these are some reactions in which pKa are implicated. How is it regulated? Is regulation of adenine cyclase is done via GS unit. I'll ask us to please mute if we are not, if we don't have the floor. try to come in with your microphones muted, please. Thank you. So what was I saying before the slight interruption? Yes, you said a regulation is either through activation of G alpha or inhibition of GI alpha, or through the direct activation of foxylin. Foxylin is used in research to strongly activate adenylyl cyclase. Now, we've seen that adenylyl cyclase is majorly reacts with pKa. How are signals amplified? On the receptor surface, on the cell receptors, activates many G proteins. Cell surface receptors are the site for activation of many G proteins, and each G protein activates adenylyl cyclase, therefore producing many cyclic AMP molecules. And each cyclic AMP molecules activate pKa, which phosphorylates many target proteins depending on the need of the body at that moment. Hours, we've been talking about activation. So once the enzyme has elucidated its effects, how is this signal turned off? Through phosphodiesterase, PDEs. Now PDEs are the ones that break down cyclic AMP and stop signals quickly. Also, in addition to PDEs, we have protein phosphatases. This protein phosphatase removes phosphate groups added to pKa, thereby returning target proteins to their original state. So I'll come again. We said once the enzyme has done its work, it's just like an artist. An artist, once he has finished performing, he must leave the stage. He doesn't keep on stage. So once PK has elucidated its effects, is now deactivated either through PDEs or protein phosphatase. Okay, and this um, is the mechanism by which the cholera toxin works. You know cholera. Who can know about cholera? What do you know about cholera? Is it Lassa fever? They say it's out now. But what do you remember about cholera? Anybody? Nobody. You've never heard of cholera? Who wants to help the class? Okay, but it's good for us to note that the mechanism we've just described about cyclic AMP is the same mechanism the cholera virus uses. Since it is similar to what we just discussed, I will go straight to the next um, second messenger for the sake of time. Okay, so this is still talking about PDEs. And we said PDEs were what again, please? What are PDEs? You can remind what is PDE? Are you sending your answers to the chat box? Hello class, I hope you are still hearing me because I'm not getting any response. Are we there? PDEs are phosphodiesterases. Okay. Okay, thank you. How do they do? What is their role? What is the role of PDEs? Okay, so just we can see we say PDE stands for phosphodiesterase, and is an enzyme that hydrolyzes cyclic nucleotides. PDEs, they are responsible to hydrolyze CAMP or CGMP in their inactive forms. So caffeine and tauphylline, non-selective PDE inhibition, is telling us how when you caffeine is in coffee, we see that in coffee, how the use of coffee promotes alertness. That's the mechanism that we just saw. Let me go back there. So this is caffeine and theophylline non-selective PDE inhibition. Okay, so methyls and things, they are pure alkaloids, they are purine alkaloids, can fit into the nucleotide binding sites. And don't forget that we said these PDEs, they are a they are a tetramer. That is, they have both um, catalytic and regulatory subunits. Okay, so what happens is that under normal conditions, cyclic AMP binds to PDE, and once this happens, you have the 5' CAMP act activated, which terminates signals. Now, in the presence of caffeine, when caffeine is present in the system, PDE is inhibited. And what happens when PDE is inhibited? You have an accumulation of cyclic AMP, which leads to PKA activation. Once cyclic AMP is accumulated, it triggers the activation of PKA, that's protein kinase A, and this leads to prolonged downstream signaling, which helps to keep alert. You know, once you take coffee, your brain, I don't know how many of you take caffeine, either in black, either in chocolate or coffee. You know that once you take coffee, you are mentally alert. Okay, so caffeine and tauphylline are non-selective PDE inhibitors. That is, they inhibit multiple families of PDE, and the families are PDE 1, 2, 4, and 5. PD3 is for inhibition of cardiac and adipose muscles. PD4 is for the inhibition of brain and immune, while PD5 is for requires much higher concentration, is a weak inhibition. So this table is showing of the effects, physiological effects of cyclic AMP elevation. If cyclic AMP is elevated, which part of the body is affected? Take for example, if cyclic AMP is elevated in the CNS, what is CNS? What does CNS stand for? The central nervous system. The effect is weakness, alert, wakefulness, like we saw in that mechanism of reaction. Wakefulness, alertness, reduced fatigue. And that's exactly what caffeine does to you. Those of you that love coffee, if it is accumulated in the cardiovascular system, you have mild positive entropy tachycardia. Okay, so I'll leave you. The table is self-explanatory. I'll leave you, but I'll touch on the last part, the diuretic. Accumulation of cyclic AMP leads to increased urine output. That's what diuretic means. It leads to increased urine output. The more of tea or coffee you take, the more you have to pass urine because it increases the renal tubules. It allows um, release of sodium or water handling. It allows the renal tubules to release sodium and water, which is why there is increased diuresis. Okay, no, what is the clinical use of tauphylline? Tauphylline is used for asthmatic patients, people that have difficulty in breathing. It's because it's used as a bronchial dilator in asthma, in asthmatic attacks. So let's look for the next second messengers. These are multiple roles of PDE, how they work with CAMP, CGAMP as a second messenger. Okay, so the next second messenger, but before I go there, okay, this is still talking about cholera and caffeine using cyclic AMP, are the phosphoproteins. The next second messenger I'll be touching on are the phosphoproteins. And phosphoproteins are proteins with a phosphate group attached to them. And examples of this type of proteins are serine, threonine, and tyrosine. Why are they important? Why are these proteins important? And like we know, but these proteins, now I want to ask us this question. Serine, threonine, and tyrosine, what type of amino acids are they? Are they essential or non-essential? In front of each, you put serine, essential or non-essential. What type of amino acids are these proteins? Serine, threonine, tyrosine. What type of amino acids are they? You have just 1 minute for this exercise. Serine, is it essential? Threonine, is it essential? Tyrosine is. When the time changes, I'm going to continue. This is 7:50. Let me harvest your responses. Let's see who gets it, who doesn't get it. Yes, great. Serine is non-essential. Threonine is essential. Tyrosine, do I agree with you on tyrosine? Do I agree with Samuel? Are you sure it's non-essential? Okay, tyrosine is non-essential. So we are correct. Yes. So these amino acids have, what did you say they have attached to them? They have a phosphate group attached to them. And why is this important? We said the phosphate group is responsible for phosphorylation reactions. And phosphorylation reactions are reactions that activate a substance. So this phosphorylation acts like the molecular switch that either turns on the protein or turns it off. So let's see one or two things that these kinases do. Kinases like pKa, pKc, they add phosphate using ATP, and phosphatase removes phosphate. Okay. Now, what is the effect of phosphorylation? Like I said previously, phosphorylation reactions are reactions that activate some enzymes. They also inactivate other enzymes, like the glycogen synthase is inactivated when phosphorylated. Also, phosphorylation reaction changes the protein shape. Phosphorylation reaction changes the protein shape. That is, it alters how the protein works because once the structure of an enzyme is changed, it loses its function. Okay, so the next is the location. It changes location in the cell. An example is the transcription factor that moves from cytosol to nucleus. Um, in protein synthesis, I don't know if you've done that, or I don't know when you'll be doing it. Also, phosphorylation creates binding sites. It creates binding sites in the cell for special domains. Okay, now the next second messenger we're talking about are the PDEs. PDEs, we've said initially, they help to break down cyclic nucleotides, CAMP and CGAMP, and they work majorly with PKAs. We saw that when we were talking about CAMP. So they are intertwined, they work hand in hand. PDEs are regulated via the calcium calmodulin pathway, also through phosphorylation, and they are also regulated locally by being anchored in specific parts of the cell, either um, AKAP, A kinase anchoring protein, using the A kinase anchoring proteins through compartmentalization. Compartmentalization is what you see in the animal cell. If you remember your animal cell wall, animals, what's that cell that we used to draw? Plant cell and animal cell. You know, the animal cell is more compartmentalized, it's broken into various parts. So that's what compartmentalization is all about. And the A caps are localized in some parts of the cell, not in all parts. And what do? We said the PDEs help to what? What do they do? What do PDEs do? They help to prevent signals from staying on too long. They allow temporal control and spatial control. They also fine-tune the strength and duration of cell response. Okay, so just like we saw in the cyclic AMP, PDEs also are critically used. What are the importance? We saw caffeine. You remember caffeine and tauophyllene? They are non-specific inhibitors of PDEs and they increase CAMP. And when this happens, we said the person is more mentally alert. And then the sildenafil, Viagra. I'm sure a lot of us have heard of Viagra. How does Viagra work? Viagra works by inhibiting PDE5, which increases CGMP. Once CGMP is increased, the smooth muscles are relaxed. And once the smooth muscles are relaxed, you have the erectile, um, it's used to treat erectile dysfunction. You also have the Rolifram. Rolifram is used in research to treat depression or inflammations. Inflammations are signs of inflammations are the arthritis. So the Rolifram are used to treat inflammations and depression because PDE, the major role of PDEs are to increase cyclic AMP. Okay, Any questions so far? Any questions so far? Okay, so the next second messenger we're talking about are the protein phosphatases. And the protein phosphatases are enzymes that remove phosphate groups from proteins, from phosphoprotein. Okay, they act as a counterbalance to protein kinases, and you have the pKa and pKcs. Okay, this is like a repetition of what I just said. Wow. Okay, this is just what we just explained. Now, any questions? So today we've been able to do a adenylate cyclase system, G protein, guanylate cyclase protein kinases, phosphoproteins, and phosphatases and phosphodiesterases. Did we touch on phosphodiesterases? Any questions, class? If you have questions, drop them in the chat box, please. Or you raise up your electronic hand. Okay, you say yes. So we've touched on, so we've done justice to today's topic. Now, the yes ma, is that a question? Okay, in the absence of questions, I'll not, it has been a long day. I'll just there is a I'll send the material to the group chat, and there's an assignment there. I don't know how soon should you submit the assignment. You have to draw some the structure of you have to draw some structures. Let's see, should I give you, today is Wednesday. By the when are you in school? When do you usually are you having any practicals this semester? When do you often come to school physically? Course rep, is the course rep around?
Hello, ma. Yes, ma. We don't have any physical class this week till next week Wednesday, ma.
Okay. What time on Wednesday is your physical class?
I think 2 p.m. to 4 p.m.
Okay, so it will be fair if I say latest by 2 p.m. next week Wednesday, you submit your assignment. Or should I
Yes, ma.
say or should I say 12 noon so that it doesn't disturb your class?
We'll submit it on Tuesday, ma.
Are you in school physically on Tuesday?
Yes, ma. We have physical class too on Tuesday, but it's not sure to hold.
It's not sure if it will hold, but the one on Wednesday, you are sure it will hold.
Yes, ma.
But you people are in the hostel now, most of you are in the hostel.
Yes, ma. That's why I said Tuesday, ma.
Now, these structures, you are going to draw it because the structures, when you draw them, it sticks better. I don't want printed structures. You draw them and you submit, because when you draw it by yourself, you are also learning. But if I say print, you just copy and paste, and I don't want that. Biochemistry is all about structures and formulas. So you will give me the structure of these second messengers. You give me the structure of second messengers and how they work. You meet the assignment in the material that we used today. So thank you, course rep Olukotu. So in the absence of any questions, Please let's all unmute and share the grace. And we'll meet this same time next week. Please let's all unmute and share the grace. May the grace of our Lord
But
Jesus
I was
Christ,
just
The love of
the
Jesus Christ,
love of
and
God
the
and the praise.
Jesus now forevermore.
forevermore.
Amen.
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 thank you for your time. See you next week. Bye.
Thanks so much.
Ẹ kúrọ̀lẹ́ mà.